hi, a very happy good evening. So this is Krishna Ma'am and we're talking about the story of life on earth, and that's biology. So thank you so much for joining my session this evening. So we're going to talk about biotechnology and applications part 4. So thank you so much for joining my session today, and please don't forget to like, share and subscribe. So I hope you guys are doing well and you guys are also enjoying your online mode of classes, so from the comfort zone of your home. So we are done with biotechnology principles and processes. Now we're talking about biotechnology and its applications. So we're already done with how biotechnology and its application is used in the field of agriculture, and to some extent how it is used in the field of medicine. So today we'll be completing how it is used in the field of medicine. Hi Nishrag, after a very long time you're here. A very happy good evening. Hi Dablu, a very happy good evening. Thank you so much guys for joining my lecture today, and I have a very fantastic news for you this evening. Oh, you told that today will be mentee, but the problem is I haven't completed the lecture. That is why there is no mentee today. That's why I never even announced that mentee is there. I'm so sorry. Once I finish this chapter, definitely we'll have a mentee. But apart from that, I have a happy news. You will have your chemistry lecture starting from Tuesday. So you will have Mr. Abhay, who will be teaching you chemistry. He will start from 11th standard chemistry. So his classes will be on Tuesday. So probably in my Monday session, sir will come and probably I'll introduce sir to you. So he will teach you chemistry from Tuesday. So in a week you'll have 2 lectures of chemistry so that you guys can benefit. So it will be completely in English again. So they're going to start from 11, so it's going to be pretty easy for you. So I wish you guys come and watch the lecture. Please do lend us your support to like, share and subscribe so that we can build up this channel. See, on a free platform you're getting so much of efforts, so please do support us. All right, so probably read karega. No, they will teach you. So they sir will teach you the basics of 11th chemistry, and then he will teach you each chapter slowly. So for now he's coming on Tuesday. He will be teaching you on Tuesday. So in Monday's class I'll introduce sir to you so that it's easy for you. All right, so a lot of people were asking me, ma'am, please bring chemistry and physics. So as of now I'm grateful that I'm able to help you with chemistry. So you'll have chemistry lecture here on my YouTube class so that it's easy for you. All right, yes. So was it really a good news? Because for the last 1 year only bio class has been going on, and so now for a change we're gonna have chemistry as well. So sir is gonna come and teach you chemistry. Yes, yes. So in the same channel where you watch my lecture, you're gonna have chemistry lecture as well. So that will be from Tuesday. All right. Yes, so let's move into biotechnology and its application. So part 4. So we have been talking about how biotechnology is used in the process of or in the field of medicine. So we saw how it's being used in the production of recombinant DNA technology using your insulin. So before we go, I have an announcement to make. So there is a session by SM sir. So your NEET counseling is going on. So I hope you guys know, for all those who have given your NEET exam once, so you should be knowing it. So there's one session conducted by SM sir. So it is in NEET Hindi channel. So it is in the other channel, not in our channel. So it's an open session. So it will be conducted very soon. So it is a ask me anything session. So any doubt based on counseling you can ask SM sir. So please keep this as your reminder so that whenever this session happens, so you can come and you can ask in your doubts, because basically based on counseling you have a lot of doubts, so you can ask. All right. Yeah, so what we'll be talking about, we were talking about the field application of biotechnology in medicine, right? So how it is being used in the field of agriculture, we are done. So when you talk about the application in the field of agriculture, so we already know so in the field of agriculture you have your Bt cotton and you have your RNA interference being used. So RNA interference is used to treat your tobacco plants, right? So these two are done. So now we are going to talk about how it is going to be used in the field of medicine. So this also we have already started. So how it is used in the production of insulin, I taught you, and how it is used in gene therapy, we are already done. So today we are going to talk about how it is going to be used in the field of diagnostics. But before that, we are going to discuss how insulin production is done. So insulin is very important because insulin is a peptide hormone. So this peptide hormone has two chains. So it has chain A and it has chain B, and both the chains are attached together by disulfide bonds. But this is the active insulin. So this is the active insulin hormone, but it also has a pro-insulin stage. So where you have a C stretch as well. So though I have my chain A and chain B, there is something called as a C stretch in the pro-insulin stage, and then we have the active insulin. Right? So what is the function of insulin? So insulin reduces blood sugar level. So that is the function of insulin. Fine? Yes, Keshav, hello, hi, after a very long time you are here. I hope you're doing really well. Yes? So thank you for remembering me and joining me in the session. So please do like, share and subscribe. Yes, ma'am, a very happy good evening. So thank you for joining my session. So please do like, share and subscribe. No, insulin beta alpha cells secrete glucagon. Yes. So your islets of Langerhans of pancreas. So in pancreas, you have a group of cells called as islets of Langerhans. They have two cells. Actually, they have three cells, alpha, beta and gamma. But alpha cells secrete your glucagon hormone and beta cells secrete insulin. So insulin is secreted by the beta cells, not by the alpha cell. Okay, fine. So this is the concept of your insulin hormone. Now, a lot of people are in need of insulin because their blood glucose level is very high. So, in that case, how will I do? So, using recombinant DNA technology, I take the genes that codes for peptide chain A and I take the genes that code for peptide chain B. Then I put it inside the bacteria. So I use E. coli. E. coli is my bacterium here. So when I put this inside E. coli, so now this E. coli is known as transgenic E. coli because I am modifying its genome. Right? So now my E. coli will produce peptide A and peptide B. Right? So now I will isolate this peptide A and peptide B from the bacteria. And I will join it with disulfide bonds. So this is how artificial insulin is produced. So since the first recombinant protein that was produced is insulin, it is also known as humulin. So we have almost 30 recombinant proteins present, but 12 of them is being marketed in India, the first one being humulin. So who was the first one to start the production of insulin? So that is your Eli Lilly and American company. Right? So these are the points you should remember. So then we went on to talk about how gene therapy is a problem in today's world. So there are some people who are born with congenital diseases, that is diseases from birth. So from the time of birth they have a problem. Okay, so why do they have congenital disease? Ma'am, explain once more. Okay, so insulin is a peptide hormone, right? So this insulin has two chains, chain A and chain B. They are joined together by disulfide bond. But before the production of insulin, there is a pre-active stage that is present or a pro-hormone stage that is present. But in the pro-hormone stage, we have chain A, chain B and an extra stretch called a C-peptide. But in the mature stage, we do not have a C-peptide. So the problem to produce insulin artificially lies in eliminating the C-peptide. So that is why to do recombinant DNA technology, I am not taking the entire insulin gene, but I am only taking the gene that codes for peptide A and I only take the gene that codes for peptide B. Then I put it inside the bacteria. So the bacteria becomes transgenic bacteria now. So once I put this gene inside the bacteria, bacteria will express it. So now I isolate that peptide chain A and peptide chain B, and in vitro or artificially I combine them using disulfide bond. So that is how we get commercial insulin. Alright, yes? Which book says insulin is produced by alpha cells? Insulin is produced by beta cells. Which book says it is produced by alpha cells? Probably it is a typo error. Please check it, okay? So gene therapy is a problem when some people are born with the defect itself. So the defect can be a genital defect when they do not have a particular gene. So probably some genes are deleted. So one example that I can give you of a congenital disease is adenosine deaminase deficiency. Adenosine deaminase deficiency. So the moment you look at this term, adenosine deaminase, adenosine is a nucleoside. Deaminase, it ends in ASC, right? So ASC means an enzyme. So when I say deficiency, it is not there. So this is a disease which is caused by the loss of this gene which codes for this enzyme. So in this disease, the gene that codes for your adenosine deaminase enzyme is deleted. Adenosine deaminase enzyme is deleted. So that is why it causes this ADA disease. Okay. So now, how do I get it right? So in that case, I use gene therapy. Actually, this enzyme is used in the production of adenine, which is a type of purine. So if adenine is not produced, or if this purine metabolism does not happen, your process of DNA replication will not happen. Alright? So that is why this enzyme is important. So now, how do I treat patients with these type of disorder? So I can do a lot of things. I can directly inject the correct enzyme. Okay, like I am injecting something, so I can directly inject the enzyme, that will work. Or I can do bone marrow transplant. So if I do bone marrow transplant, what will happen? Bone marrow is the precursor which produces your blood cells, right? So here itself, it will get altered. I can directly insert the gene, so every single cell will have the ADA gene. But this is not successful, and this requires repeated interval of, what to say, of injection of that enzyme, right? So I have to continuously inject this enzyme after quite a few intervals, and this is not very successful. So what can I actually do? Though these two are not for long term, I can still use recombinant DNA technology here. So I take the lymphocyte. So this gene therapy was tried on a 4-year-old girl. So I isolate the lymphocytes. So lymphocytes are a type of A granulocytes. A granulocytes of your WBC. WBC has two categories, granulocytes and A granulocytes. So A granulocytes of WBC has your lymphocyte. So this lymphocyte is taken, and I am culturing the lymphocells. So I am allowing it to grow artificially in lab. So meanwhile, I take a functional ADA gene. So meanwhile, I take a functional ADA gene, and I clone it inside a bacterium. So this is my functional ADA gene, and I have cloned it inside a bacterium. So since I am going to give it inside a human, I use a retrovirus. For plants, we use TI plasmid, and for humans and animals, we use retrovirus. So inside the retrovirus, I have the viral genome. But I take this viral genome, I delete this viral genome, but instead I insert my functional ADA gene. That is why it is used as a vector. Now I insert this functional aDNA into the lymphocytes that I have grown. So my lymphocytes will now have the functional ADA gene. Yes. So this is my lymphocytes with functional ADA gene. So now I will inject this into the child bag. So the child will now have the proper enzyme? Yes. But you should understand your lymphocytes, which are basically your B cells, are not immortal. They do not live always and they have a specific lifespan. So now I have to keep repeating this process every now and then. Again, it is not a permanent solution. Then what should I do? I should introduce this ADA producing gene inside the stem cell of the child so that this is a permanent process. Okay, so these are the two mechanisms, or these are the two applications that we saw in the field of medicine. But we have one more application as to how biotechnology is used in the field of medicine. So let's talk about that. So it is used in the process of molecular diagnosis. So whenever you go to a doctor, sometimes the doctor says, so normally we have general fever and we have body weakness. So the doctor gives you few supplement tablets and an injection to boost your immunity and you're fine, right? But sometimes the doctor says give blood test, give urine test. Because he suspects a pathogen is growing, he suspects a bacterial disease is there, a viral disease is there, so on and so forth, right? So he is not pretty sure, so he wants to do a further diagnosis. So he goes for a blood test or a urine test, right? But blood test and urine test are not very accurate to detect certain disease. So you would have heard some cases, for example, a person said he has only headache, but when he went, he had cancer. A person just had fever and the next day when he goes to the doctor, they say his kidneys are failed, right? So anything is a mild indication of a complex thing. So why didn't this indication come much earlier? So why are we not able to detect things much earlier so that we can give treatment? So till today, we are not able to recognize cancer in the initial stages. Very rarely we recognize cancer in the initial stages. Mostly it is recognized in the stage 3 or stage 4, which is very, very fatal. Right? So then how can we diagnose it earlier? So here is where your biotechnology comes into picture as to how we can do early diagnosis. So effective treatment of the disease includes early diagnosis and understanding its pathophysiology. For example, immediately if I detect this is stage 1 cancer, I know in stage 2 where it will go, right? I can stop it, right? So I can use some deduction techniques to detect it in stage 1. So for that, I use biotechnology. So using conventional methods of diagnosis like blood and urine analysis is not possible. Early deduction is not possible. So here comes your recombinant DNA technology, or you can use your polymerase chain reaction and ELISA. So these are some of the ways by which you can do early deduction. So using recombinant DNA technology, you can do diagnosis. Using polymerase chain reaction, you can do. Using ELISA, you can do. So we already know what is polymerase chain reaction, right? Conventional means the normal method. So normally, age-old time, how do we detect? Blood and urine sample. So conventional is the normal method. Alright. So we are going to talk about PCR and ELISA, but PCR we discussed in the last chapter. So probably I'm not going to discuss now, but I'll give you a quick recap. But first, let's talk about ELISA. So ELISA is an antigen-antibody technique. So don't get confused by looking into the picture. First, I'll explain, you understand, and then we'll look into the picture. So we are talking about ELISA. ELISA is a technique. It's a diagnostic technique. So ELISA stands for enzyme-linked immunosorbent assay. So this is the full form of ELISA. So why do I do ELISA? Is to check the presence of a virus or not. Is to check if a virus is present or not. For example, I have patient A and I'm suspecting that this fellow has HIV virus. I am suspecting. A person who is tested positive for HIV virus need not be suffering from AIDS. HIV is a virus and prolonged infection of HIV leads to AIDS. For example, 10 years or 7 years, if I have HIV infection, then that condition is called as AIDS, alright. So, otherwise, if a person has 1 or 2 HIV virus, it does not mean he has AIDS, alright. Fine. So, I am suspecting patient A has HIV virus. So, how will I confirm it? Confirmation is very important, right? So, I take the blood or the serum sample of patient A. So, serum is without clotting factors, right? So, blood has the clotting factors. So, I take the blood or the serum sample. So, if the person has HIV, in the blood, the HIV virus will be there, right? So, HIV virus is foreign to the body. So, if it's foreign to the body, we call it as antigen. So, antigen refers to any particle that is foreign to the body. Okay? Yes, Krishna Prasad, I am teaching you ELISA test. Yes, thank you so much for joining my lecture today. So, please do like, share and subscribe. So, anything that is foreign to the body is your antigen, right? So, when an antigen enters your body has a mechanism of killing the antigen. Your body kills the antigen by producing what is called as antibody. So, antigen is universally represented as Ag, antibody is universally represented as Ab. Fine? Okay. So, when I suspect patient A is having HIV virus, I take the blood or serum sample of that patient. There is a plate that has 96 wells. So, I have a rectangular plate of 12 into 8, 12 8s are 96, right? So, 12 into 8, so I have 12 rows or 8 columns, right? So, I have depressions like this. So, these are called as wells. It looks like wells, right? So, like this, I have 96 wells, okay? So, whenever you're doing a live sample, whenever you're doing a live analysis, you make sure you have it in replicates, because you do not have to do a false confirmation. So, this is something you're dealing with a person's life. I cannot do it in one small sample and confirm it. I have to repeat it so that my error is not being highlighted here. Okay, so because handling error also happens, so that is why I am taking many replicates. Okay, but only if undetectable for so long, only it leads to AIDS. Suppose if they detected it early, suppose they had HIV for more than 20 years living normally by taking medication, then they don't have AIDS. Yes, you're right, okay. Matrix aa gaya? No, it is not matrix. I'm not going to teach you maths, don't worry, I'm just giving you the idea. Yeah. Yes, Keshav, you're right. So, I take the blood sample of the patient and add here. So, first I take the blood sample of the patient and I add in all the wells. But I don't know if the patient has HIV or not. That is what I am going to test. So, inside the plate I am adding the blood sample, right? Suppose if I add the blood sample and if the blood sample has my HIV virus, I mention it as antigen. Now, from the top, I add primary antibody. So, whenever an antigen is there, the antibody will go and bind to the antigen. So, antigen and antibody complex has to be formed. So, when an antigen enters your body produces antibodies. Right? So, immediately your antigen and your antibody will bind. That is how your antibody kills your antigen. Suppose when I add the serum sample here, if the person has the HIV antigen, then when I add my antibody, primary antibody, the antigen and the antibody will bind. It will form an antigen-antibody complex. But I cannot see it with my naked eye. It is microscopic. So, I add a secondary antibody which is attached with an enzyme. Now, the secondary antibody will come and attach to the primary antibody. So, primary antibody is the antibody to kill this antigen, right? So, suppose if I have an antigen, if my HIV virus is an antigen, my body will produce an antibody. So, that is my primary antibody. But here, for my diagnostic purpose, I am also producing a secondary antibody which is attached with an enzyme. I will tell you why. Suppose if the antigen is there, primary antibody will bind. If primary antibody is bound, or if the antigen antibody complex is there, my secondary antibody will bind. But I still cannot see anything. Everything is happening and it is microscopic. But since I have attached an enzyme here, I will add a substrate from top and I will make sure it gives me a colored product. First, I will add the blood sample, then I will add the primary antibody and I will wash. Wherever the antigen is there, the antibody will go and bind. Wherever the antigen is not there, the loosely flowing around antibody will be washed off. Now, I will add secondary antibody. Wherever there is this antigen-antibody complex, my secondary antibody will bind. Others will be washed off, right? Now, I will add a substrate. Now, when I add a substrate, if the color changes, it means the antigen is there. So, I measure the intensity of the color using a spectrophotometer and I detect the levels of HIV that is present. So, this is how, or this is why we do ELISA. So, you can see it here. So, the antigen is here. Yes, so my antibody binds here, primary antibody. So, I can draw it in any way. So, they have given it pictorically in this form. So, this is my primary antibody, this is my antigen and I have my secondary antigen which is associated with an enzyme. So, when I give a substrate from top, it is giving a colored product. So, my entire 96 wells will turn colorful. Now, it is easy for me to detect if there is antigen or antibody. Now, I will measure the intensity of light. So, this is your ELISA technique. Alright? Yes. So, now talking about PCR, though I have already discussed PCR in the previous lecture, in the previous chapter. So, I am going to brush up what is PCR. So, PCR stands for polymerase chain reaction and it is basically used for the amplification of a particular gene. For example, to test if you are COVID positive, we do PCR, right? To check if the viral gene is inserted in your genome. So, you amplify and you cross check if it is present. So, I have a gene. So, I have to amplify only this portion. Then, I will do PCR. Okay? Yes. So, PCR happens in 3 steps. One is denaturation, one is annealing, the other one is extension or elongation. So, these 2 strands will first move apart and then in the next step, the primers will come and bind and then in the elongation or the extension step, it will be extended. So, one small portion is being amplified into 2 copies here. So, first step is denaturation, second step is, this is denaturation step, this is annealing step and this is my extension step. But what are the requirements of PCR? So, you should have a template DNA which you have to amplify. You should have a primer. You should have tag polymerase which does the extension step. Plus, you should also have your nucleotides like your DNTP, which includes your DATP, adenine, guanine, cytosine, and thymine. So, all these have to be present for the process of PCR. Yes, extension is equal to elongation, but we mostly say extension, we don't say elongation. So, we follow your NCERT, so remember extension step itself. Alright? Yes, so this is your PCR. The last way in which your molecular diagnosis happens is through a probe. Right? So, what do they do in a probe? So, it can be a bit complicated. Pay attention. You are only replicating? Okay, fine. So, what is this molecular probe? So, why do we do this? Suppose I have a patient A and this patient A is not normal. So probably he has a congenital disease. That is a birth disease. So from the time he is born, he is sick. But I don't know what is the reason. So now how can I find out what is the reason? He looks different. His face looks different. So what is the problem? Which gene has the problem? So please remember, the entire human genome is sequenced, meaning the order of a nucleotide is known. So that is what we did in your human genome project. So I have all my 2.2 meter of DNA sequenced. I know in which order the nucleotides are placed, right? Now, if I doubt this person has a mutation for which I need to understand where the mutation happens. So if someone shows me the right answer key, I know where I have made a mistake, right? Amplification and replication. Okay, I understood, yes. So when someone gives me the right answer key, I know where I have gone wrong. So I need a template, right? So my template here is my human genome project where my correct nucleotides are there. Now I compare this with a person who has a disease and I find out, okay, the mutation is here, right? Yes. Amplification and replication are different. See, replication of DNA happens, but replication can happen within your body. But amplification, we do it outside your body. So we use tag polymerase, we do not use DNA polymerase. Okay? Yes. So, if I suppose patient A has a mutation, I will sequence his genome. I will not sequence his genome, I will isolate his DNA. I suspect he has a mutation in his gene. So mutation is alteration in his DNA. Okay. So I will isolate his DNA. So when I isolate his DNA, I will make sure that this is separated like this. Okay. Fine. Now, from the human genome project, I also know the sequence of a normal DNA. So, from this normal DNA, I prepare a probe. So, probe is an oligonucleotide sequence, that is, it has less than 10 oligonucleotides. It has less than 10 nucleotides. So, oligonucleotide means less than 10 nucleotides it has, which can bind here. So, it is also complementary to the strands of the patient. But this probe is radio labeled. So, the patient's DNA is like this. I have the same DNA from my human genome project, so I produce probes, small sequences which can go and bind. But this probe is radioactive. So wherever this radioactive light comes, I know it is normal. Probably I have another probe which does not bind at all. So I know the sequence of this probe, so I take it back to the human genome project and then I will say, okay, this gene is mutated in this patient. So that is how we do molecular diagnosis. So DNA sequence, I separate the DNA sequence of the patient. On the other hand, I have the probes ready. So when I allow these two to mix, the probe will go and bind to the complementary sequence. So the probe is based on my original sequence of human genome project. So wherever the probe goes and binds, it is radioactive, radio labeled, so I can easily find out. Wherever it is not bound, I know mutation has happened there. Did you understand? So this is also a way of molecular diagnosis. Understood? So how recombinant DNA technology is used in the field of medicine to produce insulin, used in gene therapy, used in molecular diagnosis like ELISA, PCR and molecular probe. Clear? Any further doubt? No? So it's interesting, no? So we are talking something live, so we are talking something practical. So I hope it is interesting and you guys are enjoying. So we'll stop here. I have only a small portion. Probably Monday I will be done, and Wednesday we can have a mentee. All right? Yes. So thank you so much for joining my session. I will stop here and I'll continue on Monday. So have a great weekend. So please go through your NCERT. Any doubt, get back to me. Alright. So thank you once again for joining me on this Friday evening. So please take care and stay tuned. Thank you.